Hello everyone, so nice to meet you. As said, I'm came from Brazil and today I'm going to present the building and breaking bridges in single scRNAseq networks. I had like this huge title, so I want to short it down, but you can still see what I'm going to talk about, drug disease, Sjogren disease, and the drug targets and how we can find them. So Chash, I'm Diego Coelho. You can find me anywhere in the net just using Diego MSCOELHO. So, and one thing that I always like to say is Coelho is rabbit, so that's why you're seeing rabbits everywhere in the slides. So I'm kind of a bit narcissistic, so I put it like a rabbit to represent the lab. So I work in Rival Therapeutics, describing and looking for targets in cancer treatment. And in parallel, I'm also a visiting professor in Brazil. So I do a bit like teaching and I advise in pro bono. So I don't necessarily work for money, but I really like bioinformatics. So I try to help the students in the PhD program that I use to work. So that's why you have like a Brazilian now looking at you, because I work still in a Polish company, Rival Therapeutics. Good. So today, the three aim that I have like for today is like try to introduce you like a concept, show one of my research topics, not a few, and also make you like an invitation by the end. So maybe you'll get like I don't know, interested in going to Brazil and trying to I don't know, work with us. Oh, good. So what's basically the concept that I want to try to introduce today? So in the last 10 years, we have like an increasing granularity of information that we have from transcriptomics data. We saw it today that people are using a lot of data as single scRNA-seq. So we went from data that would describe something mixed up, like in a smooth, and now we significantly increase our comprehension of complex phenomena by separating each cell unity separately using single scRNA-seq data. So this is quite important, this is quite useful, because before it was quite difficult to, let's say, the convolute separate what you can find inside this information. And now with single scRNA-seq data, you can start to reach and understand what is going on within each cell type. So, okay, the tool that I want to introduce you today is HTWGCNA. WGCNA comes from Weighted Gene Co-expression Network Analysis. That it's a very widely used tool for bulk RNA-seq. And recently, very recently, in the last 2 years, Morabito has described a way of correctly, let's say, or the best way to can start like, oh, the point of the word, but anyway, to start like using WGCNA with single scRNA-seq data. Because we have like a huge sparsity in this data set, lots of zeros, sparsity is basically lots of zeros in the single scRNA-seq data. So they find a way of very easily trying to avoid this problem, creating like what they call meta cells. So instead of having like each cell separately, they will aggregate 20 to 50 cells within a cell group. And after this, do what WGCNA was already doing, finding the functional correlation modules. So basically, as the figure A is showing, so we have like a bunch of different cell types. You create a meta cell of a cell type, you create the meta cell expression, and after this, you use WGCNA as you were using in bulk, and you can find like the functional modules for each cell type. What is really interesting showing now, so this is one of the findings of the main paper, that they were able actually to find with autism disorder data set functional modules in inhibitory cells that was very specific to patients with autism. So they find, for example, a specific module. Oh, it's blurred here, but I can explain still. They found like two modules, for example, the NIHA M11 and 13, that was specific to the trait of autism, and also which subcell type was related to this. So one of them was parva-albumin subtype of inhibitory cells, and they found it that it has like some markers of synaptic transmission that was altered specifically within the subcell type, and this was also correlated to the main mutated genes that are correlated to autism disorder. So this is very cool. So you can select like one cell type and specifically find a phenomena that's happening in this subcell type. So moving on, so what basically WGCNA and most of those co-expression networks can deliver to us, so they are able to create a cell type specific network that will enable us to have like a more clear understanding of what's going on within the cell. And what we can do next is try to look at those co-expression networks and try to figure out what is happening there. So what we can do if we take like one of the, what this gene represents like in this network. So it's a bit differently from the paradigm that most people use that differentially express genes. So we're not looking that much about if it's higher or lower expression, but how this gene is correlating with other genes inside the same cell type. So this is quite cool. So it's a paradigm that was used for bulk RNA-seq for a long time, and now we can have like the same basically for single cell. Because one thing that I forgot to say is it wasn't quite working the last 5 years. So this was one of the first methods, I would say, that really aimed like a way of working with co-expression networks. And what is the research topic that I want to talk today? So Sjogren's disease, this very difficult name and very different one. So what is Sjogren's disease about? So Sjogren's disease is a chronic autoimmune disorder that mostly affects women after 40 plus, I would say. So mostly related to menopause. Still not well known why it's fully correlated to menopause, but most women with menopause would have can have like Sjogren's disease. And for a long time, it was just it was not widely seen because it's the woman will have like dry mouth and dry skin, dry mouth and dry eyes. So it's like a burden. It's bad, but doesn't kill anyone. So basically, the moisturizing glands are being attacked by the autoimmune system of those persons. And for a long time, people only thought like, oh, this is similar to Zika. That's another syndrome. That's not autoimmune. That will have like the similar outcome. And this is not like in some way quite important, but they discovered also like a huge correlation with all of different kinds of disorders, neurological disorders, lung disorders, etc. So now, even B-cell lymphoma. So now they are seeing that this is not just a chronic disease that will decrease the life quality of those women mostly, but also a disease that can create another, let's say, secondary problem that would affect those women's life. So for quite some time, it was difficult to understand what's going on, but since we started getting like single scRNAseq data specifically, we started having a better understanding of what is going on. So this is one of the last revisions from Baldini, 2024, that basically it has like this communication with B cell and CD4 T cells that would influence the infiltration of other immune cell types within the salivary glands epithelial cells. So I'm not going like into the details, but basically it has a lot of handshakes, let's say, in autoimmune in the immune system that would make like this infiltration and the attack of those salivary glands effectively, and you start losing those moisturizing cells basically. And one of the things in the last 3 years that they are trying to do is try to map what they can do, like mostly in specifically the B cell with T cell communication that would start like this attack from the immune system to the salivary glands. And they found like all of this, let's say, possible targets that they could use in their communication. And one of the main ones that are being described recently is the reuptake of the IgG autoimmune antibodies. So they created a FcRn inhibitor that would be quite good for those patients. So this is one of the only drugs that's in phase 2. So you can help like those patients with Sjogren's disease. So what we can do now, what we can do next, what can be like the next step, so we can understand not just a bit better about the phenomena and what we can do to find like maybe like other targets or better targets to work on Sjogren's disease. So what we did in the lab, so this is João, the PhD students that work with me. So we have like 4 data sets for Sjogren's disease. 31 patients, 18 control, 31 patients, 18 control. We integrated the whole thing using Sura. That's probably one of the most used single scRNAseq data tools. And after this, we created the functional modules using HDWCNA. And we had some other steps, but basically researching drug bank and Campbell, what we can have as already drug that can affect like the system and will be like nice to use to treat like children's. So this is like the UMAP that we found. This is just to show like all the cell types that could be found. So in the left side will be like the peripheral blood main cell types, and in the right side the salivary gland ones. We have like endothelial cells, pericytes, fibroblasts, etc. And in the left side, the ones from immune system. So this is like one of the few findings that I'm going to show you today. That basically this is like hub genes that we found for monocyte M4 and M5, and what these genes can tell us about. So one thing that we found that was really cool, really interesting, was FcGRT. That's the gene that influencing the reuptake of IgG antibodies was inside this hub, the monocyte M5-1, in the right side. And as I said, this is one of the first drugs that are in a double-blind experiment that would probably be approved to treat patients. And other thing that's really interesting about like the clusters that we found was in the M41 we saw like ITGB2 that would bind, would be like a molecule that would help like glandular cells to bind using ICOM as one of the genes as a target. And keep this in mind, the ICOM1, so ITB2 with ICOM. And the ITB2 has like also already been validated to treat one of the other traits that I mentioned, that was Zika, to help in the dryness of the eyes. So this could be like a correlation. So they was using to try Zika, but they were treating Sjogren's without knowing. And the other one, that the other module was the M51. So is a the job of the M5 module was to be like moved through the tissue and engulf the antibody. So like reuptaking, as we were thinking about, since FcGRT, the one of the main targets for reuptake was there, we are thinking like this module could be like influencing in this mechanism. And CSK has like a similar mechanism, but mostly to inhibit the B cell signaling and the tolerance to self antigens. So we separated in some way the module M4 to be like the effector, that would be like the module that will make the cells capable of infiltrating in the salivary gland cells, and the processor one. So this one of the mechanisms that would help in the reuptake of the IgG. And one thing interesting that we found in parallel was we found another module in another cell type, the endothelial cells, that has actually ICANN inside. So this is the one that I showed you before. So in some way, we found a module that will be the one that makes the monocyte able to get inside the salivary gland cells, and the other module that would be like the receptor that kind of make the handshake between module M4 and the module M8 in the endothelial cells. So as already said, so it has like this mechanism that's already described that create like these physical handshakes to make it possible for the cells to infiltrate the cells. So this can be like really two good targets that are not necessarily being used still in the treatment of children, and this is also have not like been fully described in like recent publications related to Sjogren to be an active, let's say, a mechanism to avoid those cells to migrate. So as I said, most of them think about like CD8 T cells plus versus B cells handshake. So this is like maybe a novel mechanism that can be explored to treat Sjogren's disease. And this is my group, so everyone from Brazil, of course. And the one in the middle next to me is João, that works with me, so I'm presenting the work from him. And these are the other lab co-members that help me in this work. So, bardzo dziękuję. If you get interested, We are always receiving students from all over the world, so this is the invitation. So we have like some PhD sponsorships from time to time. So if someone wants to go to Brazil, maybe visit for a few months and try to do like research in Brazil. So we have like these opportunities there. You can find me anywhere with @Diego, MS Coelho, etc. And this is my contacts that you can reach me. So thank you so much. Fique.